Hello and welcome to PW English, and I'm your Diksha ma'am. So, guys, here we are going to start our next chapter of biotechnology. Biotechnology unit has two chapters. One we have already covered, the biotechnology principle and processes. So, there I have taught you about how the biotechnology works or how do you do all these experiments. Now it's time to see what are the application of biotechnologies where you are using it in the different fields, right? So here we'll be talking about what we have already made by using biotechnology, and how are we using all these products, what were the problems and what are the solutions of biotechnology has given us so far. So let's get started with this chapter. So we are going to talk about the introduction part first. All right. The biotechnology, as you would have learned from the previous chapter, essentially deals with the industrial scale production of biopharmaceuticals, biologicals using genetically modified microbes, fungi, plants, and animals. All right, guys. So this is a cutting from NCRT. So it's a first paragraph of NCRT. So don't worry if you're thinking, ma'am, from where we can read this. So this is from NCRT. All right. So if I'm talking about biotechnology in previous chapter, we have seen how at the industrial scale, how at a larger scale you can produce a products. Now, what are those products? So as I've told you, there we were producing the insulin. So whenever we are talking about biopharmaceuticals, okay, so there are two things. One is insulin, one is the pharmaceuticals, another is therapeutic. What's the difference? The pharmaceutical is like your tablet that you eat while you get sick. Fine. And therapeutics are something which are normally produced in your body, but when it's get deficient and you get disease, so those all products are therapeutics. For example, insulin hormone is a kind of therapeutic because that hormone is naturally produced in your body, but sometimes in some diseases it gets slow. Fine. So by this we can also produce by using biotechnology we can also produce therapeutics. We can also produce the pharmaceuticals like medicines and biologicals using genetically modified organisms. So here the organisms that we are using, we have modified them according to our need. In the previous lecture I have told you that we are modifying the genes. Fine, we are adding the gene of interest in the vector. This was what we were doing. So technically we are modifying that. Fine. So whatever here we are doing is we are giving gene to another organism, forming the recombination. As a result, the organism is getting genetically modified. Okay. So here the organisms that are modified can be any microbe, it can be fungus, it can be plants, even it can be animals, right? So we can modify the cells of any of the organism. The applications of biotechnology includes therapeutic, diagnostic, genetically modified crops for agriculture, processed food, bioremediation, waste treatment, and energy production. So where are we using it? This is what we are going to talk about in this chapter. So what are that fields first of all? First of all, I've told you therapeutics like we were talking about insulin. Okay. Diagnostic, to diagnose the disease, like in COVID we have done that test, right? RT-PCR. So whosoever was in the contact with the diseased person, they have to go for this test, right? That was RT-PCR. So yeah, that is also a gift of biotechnology. Then we have genetically modified crops for agriculture. We have modified the crops according to our need. As a result, the yield should increase. Then we have processed food, the food that are made in the factories, right? That's also a gift of biotechnology. Bioremediation. What is bioremediation? To decrease the pollution by using biological components. For example, you will study this in the botany. For example, whenever there is oil spills, wherever there is oil spills, there a particular bacterium are spread out, so that all that bacterium can drink or eat that oil, and the oil vanishes away from the oceans. Fine. So that's remediation. Waste treatment, plants, and energy production. So for all these things we can use the biotechnology. Now, what are the critical research areas of biotechnology? We have done everything about the research and how the techniques are done, right? So, but still, there are certain areas there which need to be researched where you want, something more should be there. Okay, so first is providing the best catalyst in the form of improved organism, usually a microbe or pure enzyme. So first thing is we should have better restriction enzyme and we should have better organisms to deal with. So if we have better catalyst, better organism, better things can be produced. Creating optimal condition through engineering for a catalyst to act. For example, you discovered an organism or the enzyme, but you don't know at which condition it can survive or at which condition it can flourish more. Like I have discovered a bacterium. I'm just giving example. I have not done that so far. Um, so, for example, I discovered the bacterium and I got to know, yeah, that's a good one, and it can go give me a million of copies of my desired gene. But if I don't know where it can grow more, whether it needs this type of nutrient medium or not, that or something like that, if I don't know about that, so it will not serve that purpose, right? So we should know that what are the optimal conditions for them. Downstream processing technologies to purify protein or organic compounds. So downstream processing is one thing that haunts us till now, because there are a lot of things that we can produce, but we don't know how you can purify them. Purify means isolating them in the pure form. You don't know the techniques to do that. Fine. For example, I have a mixture of nutrient medium and I'm producing X hormone there, but I don't know how to isolate it from that. Fine. So this is the thing. These are the three areas where you need to work more and do a lot of research and get to know another things. Okay. Okay. So moving further, we'll talk about the applications in agriculture first. So if I'm talking about the application in the agriculture, what's the need here? Why agriculture? Think. Why there is a need? Why there is a need for a lot of biotechnology in the agriculture? Very simple question, very simple answer. And that is because population is increasing and there is increase in food supply. So what's happening around there is, as the population is increasing, we are reducing our agricultural land, we are making houses and all. We have less agricultural land, but we have a lot of people to feed. Fine. Also, the soil is limited and soil get exhausted at time, or the plants, they are not that high yielding. Then to increase the yield, we need to have biotechnology in the agriculture so that there is high yield of the plant and we can eat more. Fine. Not eat more, eat according to our needs, but a lot of people, they can get that food. Okay. So what's the problem? Let's first talk about the problem. Problem is there is increase in population and there is less food supply. So to increase the food supply, we have three options. What we can do, we have three options. One, we can use the agrochemical means. That means we can pour a lot of chemicals. We can pour a lot of chemicals to our crops, like pesticide, insecticide, fertilizer. If the soil is not fertile, put the fertilizer. If there are a lot of insects that are eating the plant and that's why the plants are dying and food supply is decreasing, you can put some insecticide. But again, these chemicals are so much harmful to our body as well as to the environment, right? Also, these are very expensive for a farmer. So what do you think? Is this good? From my side, it's a big no, right? So agrochemical means can be adopted. What are these? It includes use of chemicals. Use of chemicals. What are these chemicals? Like insecticides, pesticides. Okay. So what are the drawbacks? These are very harmful. They are harmful and expensive. So what's the second option? If these are harmful and they're not good for the environment, why not to go for everything that's organic? So nowadays you must be herding things that are organic, right? If you go to the go for purchasing the pulses and all, they will ask you want organic, you want normal. Organic farming is a type of farming wherein you're not using any chemicals. It's completely nature friendly. So this organic farming is nature friendly. How? Whatever you're using to kill the pest and all or increasing the fertility, you're using everything that's natural. For example, you do not use fertilizers, you use manure. You know how manure is formed? If you know, just let me know in the comments. So all these things, they are nature friendly. Like if you're using insecticide or pesticides, they must have been extracted from any plant. So everything is nature friendly. But what's the drawback? This is very slow. Okay, the organic farming is quite slow nowadays. People are doing it, but it's not that much efficient to feed the entire population. That means you just cannot switch the normal farming to the organic as such. It takes a lot of time. Second, third thing, and that's the another option that we have, that is genetically modified crops. Genetically modified crops. That means you're using the plants and you're modifying the genes in it. And while you are modifying the gene, that means you can add new genes to the plant. You can also subtract the bad gene to the plant. So in genetically modified crops, what we are doing, we are modifying the gene. How? By gene addition. That means you can add new good gene, or you can also, if the gene is bad, you can also remove the gene. So by doing this, we can modify the crop, and you modify the crop according to your needs, definitely, right? So here you can use these plants. How they are better than the other? Let's see how these are the good option, how they are better than the others. The genetically modified organisms or the crops, they are better in a way. Why? They have made crops more tolerant to abiotic stress. So apart from the biotic things, there are certain abiotic stress that a plant faces. What are those? Like drought, that means there is no water, there is a lot of cold, there is a lot of heat, or their soil is quite salty. The plant can die easily. So what if I made a plant or I add a gene that make it resistant to all these things? The plant can easily grow, right? So we have made the plants in a such a way that they are more tolerant. They are more tolerant to the abiotic stress. Second thing, the second problem a plant faces is the pest, right? These pests, they are like, they can eat all your crops and they will leave nothing behind. So it's a big no for you, right? You don't want your plants to get die and everything gets into the losses. Okay, so what we do, we have made certain plants, which are pest resistant. That means the pest cannot harm the plants. So by that, there is a less dependent of plants on the chemicals, the agrochemicals that we were adding, right? If I made the plant itself which is resistant from the pest, the plant can protect itself from the pest. You don't need to spray a lot of insecticide. So it's a save of money, it's a save of time, and a save of the environment and your body, right? Then help to reduce post harvest loss. What are post harvest loss? Harvest means cutting off the plants after they get mature. So after you pull off the plant, or for example, we have tomato, after the tomato gets mature or ripened, you just take it out and you store it so that you can sell it in the market. But do you know, these tomatoes, they get so much softer after 3 or 4 days. But if a farmer is living in an area and the market is around very far away, fine. So it's all the tomatoes, they're going to, get stale. Okay, so what if I made a crop? What if I made a crop where their fruits or the vegetables shelf life gets increased? Shelf life means the time duration up to which you can keep it safely. It will not spoil. So what if I made a plant in such a way that its shelf life will increase? That means the post harvest loss, that means the loss you are getting after the harvesting of the vegetable or fruits, that can reduce. Okay, so see how many benefits the GMO have. Increase efficiency of mineral uses by plants. If I know there is a soil where the minerals are very less, okay, or I am adding the minerals externally, I am adding a lot of it. So if I made a plant that can survive itself with the less mineral demand, it has a very less mineral demand, pretty good for you. So you can also make plants which have, less mineral usage, and or in the less mineral it can efficiently work. Enhanced nutritional value of a food like golden rice, vitamin A enriched rice, which we'll be discussing. What does it means? Enhanced nutritional value of food. You can also increase the nutritional value of a food by adding something. For example, if I know this area's people, or in this area the crop X can grow, no other crop can grow, and people are dependent on X crop only. Okay, but that crop or that food doesn't have enough vitamins. What if I genetically modify that crop and add a lot of vitamins in it? The people will get it, right? Because they're eating that food. They are, they have no other options. Fine. So by this, I can increase the nutritional value of a food. But why GMO? You must have heard of Green Revolution. Yes, you must have heard of Green Revolution. Green Revolution, it is stated that by Green Revolution came where the food supply was increased by using different techniques. It is stated that Green Revolution has increased the food supply. It actually tripled the food supply, right? So if you know the Green Revolution has already tripled the food supply, then why are we using GMOs? You can use all the things the Green Revolution was using, right? The thing is, the Green Revolution has used better variety of plants. Okay, how they got success? Because they use better variety of plants. Second, they used a lot of chemicals. That's why that's a gift of the Green Revolution. Nowadays, they're using a lot of chemicals, right? And third, they show or they taught the farmers about the management of the plants. They taught the proper management skills, like if this is a crop X, this will need this amount of water. At this time duration, you have to give water. In this season, you have to grow the crop. This is the fertilizer you need. So all that lead to the success. But again, there are a lot of hindrances or the negative points of Green Revolution. That's why nowadays they are saying that you should use GMO. And what are these hindrances of Green Revolution? Hindrances or drawbacks. First hindrances is these agrochemicals are itself not good. As I've told you, these agrochemicals, they are harmful to us, to the nature, and they are so much expensive that a farmer will not be easily able to afford. Okay. Another thing, the plant variety they are using, it was capable to grow in a particular in a particular regions only, in particular continents only. For example, the wheat variety we are growing, it will not be a great success in the colder regions like Alaska and all, right? So the varieties of plant was for a particular continent, right? So you know about lot about the Green Revolution now, and you have also, I have told you about the GMO or GMC, genetically modified crops. What do you think, which is a better thing? Definitely genetically modified crops. So that's why we can use these crops, and with the less money, using less expenses, we can grow better crops which will fulfill all the purposes. So if I'm talking about the golden rice, what were these golden rice? So in some regions of India, people are dependent upon rice. Their staple food is rice. Okay. So it was find out that people who had the staple food rice, they were suffering from two deficiencies. One was vitamin A deficiency, and second was the iron deficiency. Okay, so let's talk about the vitamin A deficiency only. So they thought if they are having vitamin A deficiency, the people were getting the night blindness. Vitamin A deficiency causes night blindness, right? So the scientists, they came up with the one different variety of the food, and that's the golden rice. What they did, they took a plant which is Oryza sativa, which is a temperate normal variety people used to eat. Okay, that's the rice. So this was a variety people were eating. Okay, so now what scientists did, they took another plant, that is Narcissus pseudonarcissus. This is daffodil. Okay, so that's the daffodil. You must have heard of the daffodil flower, I guess. So they took a beta carotene gene. They took a beta carotene gene from the daffodil. Now you are ma'am, you are saying these are vitamin A enriched rice, and now you are saying that we have taken the beta carotene gene. See guys, there is a mechanism in our body. The beta carotene, which is rich in the carrot as well, that's why we say carrot is good for eyes. The beta carotene is present in that. So when you eat beta carotene, it's a precursor of vitamin A. It goes to your liver, and in liver it gets converted into vitamin A. So we say beta carotene is a precursor of vitamin A, right? So I'm writing gene here, but who is a precursor of vitamin A? The beta carotene, not the gene. Okay, so we have taken gene of beta carotene from this plant, and by using Ti plasmid, we added this gene into Oryza sativa. So what we have used? Ti plasmid. If I'm using Ti plasmid, what will be the bacterium? The agrobacterium. So that's the vector we have used, Agrobacterium tumefaciens. And by using this vector, we added this beta carotene gene taken from daffodil into the Oryza sativa. So now, since it is beta carotene gene, so the grains of this rice, because you have genetically modified it, so the grains of this rice, they appear golden in color. So that's why you call it as golden rice. So now Oryza sativa have this gene, and this will become a golden rice. Why you call it as golden rice? The grains are golden in color. Are golden in color. All right. Okay. So if someone asks you what were they enriched of, they were enriched of vitamin A. So now if people they eat enriched with vitamin A, if the people they eat golden rice, they will not feel the deficiency of, they will not face the deficiency of vitamin A, and hence no night blindness. All right. So this was one example how we have increased the nutritional value of the food by using genetically modified crops. Talking about next, you must have seen this in the NCRT. I hope you have seen it. That's the BT cotton, right? So now the next thing that we are going to talk about is pest resistant plants. The plants or the crops pest resistant means they can fight against all the pest. What is a pest? Pests are usually insect that kills your plants. Okay, they live in your plants, they eat them and they destroy them. For example, if you can see, here is a normal cotton ball, and here the cotton ball has been destroyed by the pest. So these two are destroyed by the pest. So the normal cotton, when you grow, the pests, they are like so much killer to them. They can easily destroy entire field. So it was a bigger problem because cotton is so much important to us. We wear clothes that's made up of cotton, right? So here they came up with one plant which is genetically modified. That's BT cotton. That's BT cotton. In BT cotton, BT stands for Bacillus thuringiensis. That's a bacterium. That's a soil bacterium. Okay, now what about this bacterium? Let's talk about everything in detail. So first of all, for understanding this, let's talk about what are the enemies of our cotton. Okay, so cotton was attacked by insects. Insect is a pest here. And these insects, they belong to three order of class Insecta, right? We have phylum Arthropoda, and then we have class Insecta, and then we have for certain orders in the hierarchy. Okay, so there are three orders. One is Lepidoptera, another is Coleoptera. There are certain other orders as well, but there are three orders which are the insect pest of the cotton, Coleoptera, and then we have Diptera. Okay, so Lepidoptera contains tobacco budworm, which this is the Lepidopteran. Lepidoptera is the order of butterflies and moths. So these, it's worm means it's larva. Okay, so its larva kills the tobacco plant. Okay, so cotton was attacked by insect pest, and out of all these insect pest, which is the order, which one is the order which kills your cotton, and out of these three, that's basically Lepidoptera and Coleoptera. So in Lepidoptera there is another worm, that kills your cotton. That's bollworm. And the last is armyworm. So someone asks you, what are the insects that can be a pest? So these are three type of pests I've told you. Then Coleoptera, we have beetles. They also destroy your plants. And Diptera contains flies and mosquitoes. Yes, they also destroy your plants. Okay. So for cotton particularly, we have the bollworm. What we have? The bollworm. Okay, so cotton was attacked by insect, and insects have further orders which can which are basically pests. So someone asks you out of which orders usually the pests are most common. So you will say these are the right. Okay, so now you have got to know the problem. Okay, so the problem is the cotton is attacked by the insect worms, the larvae of the worm. Now what next? What can you do? So when you have your friend cotton and the enemy is insect, so if you want to destroy your enemy, say this is you, okay, you are cotton. If you want to destroy the enemy, let's find out the enemy of insect, and that's the Bacillus thuringiensis. Okay, you are not killing the insect straight away. We were doing that by spreading the insecticide, but what now we are doing? We are finding the enemy of the insect, and that is a bacterium. Now what about these bacterium? Let's see. So we have this bacterium which is Bacillus thuringiensis, which is a soil bacterium. So whenever this bacterium, it undergoes sporulation. What is sporulation? Formation of spores. When this bacterium, it undergoes sporulation, that is formation of spores, this bacterium produces a protoxin. It produces a protoxin. This protoxin, when it gets the alkaline conditions, it gets converted into endotoxin. And this endotoxin is the one which is deadly to the larva of your pest. Okay. So what these pests do, they deposit their eggs, and eggs becomes larva, and larva feed on your crops there. So what we find out? We find out, let's find the gene that is producing this toxin. Okay. We find out this toxin, protoxin, is produced by a gene which is a cryogene. Cryogene produces a protoxin, and then the alkaline conditions convert it into endotoxin, which is basically a crystalline protein. It is in a form of a crystalline protein. From here, the crystalline CRY is used in a cryogene. So we saw that bacteria have this gene, and this gene produces toxin. Let's add this gene in our cotton plant. So what we did, we took our cotton plant, we took cryogene, and we added this in our cotton plant. By using, definitely you know that, agrobacterium. By using agrobacterium, we added cryogene. From where have we taken the cryogene? From bacteria. We added it into our cotton plant. Now this cotton plant becomes BT cotton. It becomes BT cotton. Okay. This BT cotton, it have the protoxin. Okay. Now the pest rise. The larva rise. So larva will consume this protoxin. Okay. Now see, the insect's larva, it feed on protoxin. So when it will feed on protoxin, its gut have alkaline environment, unlike that of our gut. Our gut's starting environment is always acidic. Like in mouths we have saliva, that's slightly acidic. In stomach it's huge acidic. So that's why this protoxin cannot harm us. So when the insect larva feed on a protoxin, the alkaline nature of gut will convert it into endotoxin. Now what this toxin will do? So in the midgut, for example, if this is a midgut, and these are the cells of midgut. Okay. These toxins will create pores here. It will create pores here like this. And due to creation of these pores, the water will enter. Okay. The water will enter. Now see, the toxin binds to the cells. This is a midgut. This is a midgut. The center part gut, like we have small intestine, it's like that only in the insect. So the midgut cell, the pores are created in the midgut cell by this toxin. As a result, all the water will enter into the cell and the cell will burst, leading to the lysis. Lysis means breakdown. Okay, so what this toxin will do? It will create pores in midgut. Now the pores have been created. The pores have been created. Once the pores have been created in the midgut, what will happen next? The cell burst and lysis. As a result, the larva will die. Larva will die. And your purpose is fulfilled. So it's quite simple. We have taken the gene from the bacterium, added in the cotton, made it Bt. Bt will secrete protoxin. So now when the insect will consume the protoxin, it will get converted into the active form endotoxin. So if you know we have this gene, cry gene, can we use cry gene for all the pests? Is it true? No, you cannot use the same cry gene for all the pests because the cry gene's function is specific to the plant and specific to the pest. Okay, so the cry gene, must write it, it's quite important. It's the part of this topic from where the question arises. Cry gene is specific to plant and pest. To plant and pest. Okay, for example, we have cry1AC, cry2AB, which will kill your cotton ball worm. Okay, and I have another gene which is cry1AB. This is for your corn borers. Now you will say, ma'am, corn? Can we make BT corn? Yes, we can make a lot of BT plants. So what they have tried right now, or another countries are using, BT tomato, BT potato, soya bean. We have made corn as well, right? So there are other plants like tobacco as well. We have made a lot of, like BT rice. Other countries are also using it, but in our country we are only using BT cotton as commercialized. We are not using other these edible things. Okay, there is also BT brinjal. There is also BT brinjal. Okay, so but in India we are only using cotton. Okay, I hope this makes sense to you now how the pest resistant crop has been made. Let's talk about the another issue in agriculture. Again, that's the issue of a pest. You can see, if you can see, let me know what is this. You can think I'm giving you 1 or 2 minutes. It's also given in NCRT. It's also given in the NCRT. What is this? Yes. So that's a tobacco plant's root. Okay. So what is the plant we have? Tobacco plant. Now, what is the pest of a tobacco plant? Now, it's not the insect. It's a nematode. It's a nematode. Yes, nematodes can also be the pest. It's a nematode. What is a nematode? These are Ascalmanthus, right? Nemathalenthus you must have done in the animal kingdom. Yes. So that's Melidogyne incognitia. Melidogyne incognitia is a nematode which is a pest of the plant tobacco plant. What does it causes? It causes the disease. What's the name of the disease? Root knot. Okay, if you can see here, these are the normal root of a tobacco plant, and here are the roots which are been or are being diseased or which are being attacked by these bacteria Melidogyne. And how does this Melidogyne attack our plant? It produces RNA. And RNA will form protein, and this protein will harm our plant. Okay, so this is how the Melidogyne harm our plant, by producing mRNA, which will later on during translation will produce protein, and our plant will get a disease. Okay, so we were thinking, oh my god, how we can prevent the damaging of our tobacco plant. So they came up with the one technique, that is RNAi. Okay, so here first of all you need to understand a technique, that is RNA interference. RNAi. Okay, it is, I stands for interference. So it's a normal natural cellular defense of eukaryotes. It's a cellular defense of eukaryotes. It is also present in my cell, it is also present in yourself, in all the eukaryotes. It's a cellular defense. So first of all, we are talking about the technique. Later on, we will see how this technique is used in our problem. Okay, so now we are just talking about the technique. So this technique is against one thing in your cell, that's double-stranded RNA. We like double-stranded DNA, but we don't want double-stranded RNA. Our RNA is always single-stranded, if you have read, right? We don't want double-stranded RNA. So now, whenever, what if mistakenly or by viral attack, the double-stranded RNA is formed, then what will happen? Okay, so whenever if there is any viral attack, for example, this is a virus that enters in your body, right? Our RNA gets double-stranded, or this virus is given as double-stranded RNA. So our body have a natural defense mechanism to destroy this double-stranded RNA, and that technique is RNAi. So whenever there is any formation of double-stranded RNA, one enzyme will come and its name is dicer. One enzyme will come, its name is dicer, and it will chop your double-stranded RNA into small pieces, and the name of these pieces are small interference RNA. Okay, so whenever there is a formation of double-stranded RNA mistakenly or by viral attack, the double-stranded RNA will be taken down into small pieces by your enzyme dicer, right? And this full form of these small RNAs, siRNA, small interference RNA. Small interference RNA. Okay. So now the thing has not been finished here, because these small strands can also be very harmful to you. Now what is the next thing to come here is another group of enzyme which is waiting that siRNA is formed and I will come and get activated. So siRNA activate the another enzyme, that is RISC, which is RNA induced silencing complex. RNA induced silencing complex. This is the one which is the important one, which will silence the silence the double-stranded RNA. Okay. So first, what has happened? Double-stranded RNA is formed. Dicer will come, chop it into small interference RNA. These small interference RNA will now activate this enzyme, which is RISC. Okay. So for example, if this is a RISC, this is RISC. Now what RISC does is, it will pick up one small interference RNA and will keep one of its strand, and it will destroy the another strand. It will keep one of its strand, like 5' to 3' it will keep, and 3' to 5' it will kill it. Why it is keeping one strand? To identify. Okay, for example, if I have to tell you that find this person, how will you find that person? Until or unless I give a photograph of this person, you will not be able to find it. So we are carrying a photograph and you're finding where it is that person, where is that person. Once it is matched, you will hold that person and get it to me, right? Okay. So similarly, the RISC is doing, RISC will keep 5' to 3' end of small interference RNA, right? Because these small interference RNA is having a piece of it and we want to destroy it. Okay. So wherever it will find the matching of it or the complementary of it, it will bind to it and it will kill it. For example, if it finds this DNA, double-stranded, RNA, it will find it, this 5' to 3', 3' to 5'. Now RISC has been activated. RISC will come here and now it will see, oh my god, That's a complementary. That's a complementary. Risk has found it. Now, risk is a very powerful enzyme. It will chop it into nucleotides. Okay, in fact, it will open up into the small nucleosides or even nitrogenous bases and sugar. So it was cutting it into the double stranded RNAs only, which are small pieces. But risk, what it will do? It will chop all the double stranded RNA into monomers or into nucleotide or nucleosides or even small bits and pieces. Okay, so this is how your body or your cells have the natural mechanism to kill the double stranded RNA if in case it gets formed. Now, how we are using it? So what if we activate a risk like this? Okay, our problem was the nematode was coming into the plant, and the nematode was giving mRNA, and mRNA forms a protein, and that protein kills the plant. I'm telling you again, the problem was, see, here is the problem. The problem was this mRNA. Okay, the problem was this mRNA. Okay, so now what if we activate a risk inside the plant? We activate a risk inside the plant that will identify this mRNA, that will identify this mRNA. Can it be possible? Yes, but for this you will have to initiate RNA interference. You have to start the RNA interference. Now the question is, how will you start the RNA interference? When the RNA interference is started, only then the risk will be activated, which will identify this mRNA. So for this you need double stranded RNA. Now how will you form double stranded RNA? So for this you need a gene, right? Because gene after transcription forms the RNA, and then RNA will become double stranded and RNA interference will start, and a risk will be formed that will be having one strand which is complementary to this. Yes, let's do it. Okay, so now what we did, we added gene 1 and gene 2 in our tobacco plant. We added two genes because we are genetically modifying it. This gene will produce RNAs. Okay, it will produce RNA 5 prime to 3 prime. Again, this is 3 prime to 5 prime. So one is antisense, another is sense strand. Okay, it will produce both sense and antisense RNA. So what we are doing is, whatever genes we are adding into the plant, these are nematode specific genes. What are these? These are nematode specific genes. We have to see what is the mRNA of our nematode, right? Because if you know the mRNA of nematode, only then you can find or form the risk which is complementary to it. Okay, so what we did, we added a gene which is specific to the nematode, then we produce RNA out of it. Now these RNA, they will get double stranded. Okay, these are double stranded RNA form because both are complementary to each other. That's why they are joining. So you have to take care that this gene is complementary to what? Your mRNA of nematode. And now whatever this gene have, we are also taking care that this gene will produce RNA. The RNA will be complementary to this, so that they will join and form double stranded RNA. Now this double stranded RNA, it will be chopped by dicer. Dicer will form small interference RNA, and now the risk will come. Who will come? The risk will come. The risk will keep 5 prime to 3 prime end. Now this 5 prime to 3 prime is this one, and this one is complementary to the nematode mRNA. Now nematode came. Nematode's mRNA came. Okay, 5 prime to 3 prime. Okay, so if I see this one was complementary to this, and this risk is already been activated in the tobacco plant which is genetically modified. Now this will bind to this one. It can easily recognize this, right? This risk which is already in the plant, when a nematode will infect and give mRNA, this risk will bind to it. This risk will bind to it. Okay, this risk will bind to it. Yes, So when the risk will bind to it, it will chop it into pieces, and this is how you are killing the mRNA of nematode. So by this, what we are doing, we are not letting the gene expression of the nematode, right? So that's why this technique, which is RNAi, it is also known as gene silencing. You will say, ma'am, we are cutting the mRNA, we are not cutting the gene. Guys, how you are cutting a gene? When the gene's main function is its expression, and its expression is formation of mRNA. When you're not letting the mRNA to work and formation of protein is not taking place, that means the gene is silenced, it's not doing anything. Okay, if I'm saying silence, that means I am not letting you speak, that's your function. Or if someone told me, silence ma'am, my work is to speak and let you know, okay, and to teach you. So if I'm silenced, that means I'm not killed, I'm just silenced, I'm not doing what I'm doing. Okay, so that's how you have silenced the gene, by using RNAi. So here there is a line in NCRT, by using Agrobacterium tumefaciens. Because it's a plant, what vector we will use? We will use Agrobacterium tumefaciens. So by using Agrobacterium tumefaciens, the sense and antisense strands both are produced in a plant. So in a plant, we have added gene. So these genes, in nematode specific genes, are added in plant. I'm writing it here. By using Agrobacteriums, because it's a plant. Okay, so we have added genes in such a way that they're producing both sense and antisense strands of RNA. So these are what? These are RNA. And since they are complementary to each other, they will form double-stranded RNA. And now this RNA will initiate RNA interference, and a risk will be formed which is complementary to the nematode's mRNA. So whenever the nematode's mRNA will try to enter into your plant's root, we already have a risk there waiting for it, and risk will bind and it will chop it into pieces. Chop it. Okay, so this is how you can make the pest resistant tobacco plant. Easy? Yes. All right, I hope you get to know what was it. Let's talk about another application. So this was about the agriculture. Let's step into the medicine. Okay, so now next is the application in the medicine, and in medicine we have genetically engineered insulin. That's good. Insulin we also have produced in the first chapter. So let's see how we do it, and who has done it for the first time. So genetically engineered insulin was, first of all, it was produced by some scientists, which was Banton, I guess, right? And later on industrial application, it was done by an American company, which is Eli Lilly. Eli Lilly in 1983, which is an American company, they produced the insulin. Okay. So how did we produce it, and what is the need? First of all, always I started with the need. Okay. So what's the need? The patient of diabetes, they were dying of diabetes, because there are two types of patient. One are with type 1 diabetes, and another are with type 2. The type 1 is autoimmune disorder, in which the beta cells of pancreas, which are producing insulin, they are attacked by your immune system and beta cells get destroyed. Hence, no insulin is produced. So for those people, insulin is like anything better in this world, like anything. Okay. So, sorry. So they needed insulin. So in the type 2 is like adult onset diabetes also. There also people need insulin, but in a severe cases. Okay, so that's why in NCRT they have mentioned adult onset diabetes. So both diabetes, somehow they needed insulin, and people were dying because of there was no treatment of diabetes, right. So whenever there is a severe case, a person are dying, so they take the insulin from some slaughtered animals like pigs and all, and they injected that insulin to patients. But there was side effect. The patients got allergic immune response. Why? Because there are different animals, we are different animal, the pig is different animal, so that's why there will be allergic reaction. Okay, so whenever you're taking an organ from from human itself, you get the immune response. Okay, so let's think about, we are getting the thing of a pig, definitely our immune response will work against it. So what happened, they were like, oh, now we need something which is quite positive and it will solve all our purposes. So they said we have produced a lot of things by biotechnology. Let's try the insulin. But the problem with insulin was that insulin is produced as a pro-insulin form, wherein it have three chain, A, C, and B chain. These are polypeptide chains. Okay, these are polypeptide chain. So when the insulin gets mature, both the chains they get separate, A and B. C is taken out, which is useless, and A and B they join to each other by forming disulfide bonds. And this is how a mature insulin it looks like. It contains two chain. One is A chain, another is a B chain. Let me change the color. It is quite confusing. A chain and B chain is present in the mature insulin. C is free. It is of no use later on. Okay. So these all are changes which takes place in your body after the insulin is formed, after the translation of protein has taken place, and all these changes are known as post-translational changes. What are these? Post-translational changes. Okay. So let's see what was a need. First of all, repeating it again and writing. Need is production of insulin, human insulin. So they have given the name humulin, human insulin, for diabetic patient. Okay. What was the earlier treatment before this? Pig insulin was used. But what was its side effect? Allergies. Allergic to human. Okay. So we want to produce it in a bacteria. Now what's the drawback? The drawback is that insulin is produced in proinsulin form. It later on gets converted into insulin, which is a mature insulin. And here it is formed during post-translational changes. But bacteria is a prokaryote. It cannot undergo these changes. It cannot do this process. It can produce it up to this stage, but this thing, the bacteria cannot do it for you. So now we have to produce it in bacteria. We have no other option. Okay. So what will we do? What will we do? So they come up with one idea. What they did, they took two genes. One is gene 1. This gene 1 will form a polypeptide chain. It will form a polypeptide chain. Then they took gene 2. It will produce B polypeptide chain. So they actually subtracted the gene. They removed the gene of C chain, right? They removed the gene of C chain. They only took that segment of gene which were producing A and B, and they took it separately. And now they added it in PBR322. And this PBR322 is conjugated with LAGZ gene. So that means you are doing selection with blue-white colonies. Okay, again here PBR322 is used. The same PBR322 conjugated with LAGZ. Once you conjugated, you joined them. What is formed? What is formed? This gene is added in PBR322. What is formed here? Recombinant plasmid or DNA. Now you added it in E. coli. After the transformation, it is added in E. coli. Now this E. coli, this one, will produce A chain. Same steps here, and here it will produce same as the other. I'm writing here. It will produce B chain. Now what we did later on, we combined these two chains, A and B. We combine these two chains. This is A chain and this is B chain. And let them join. And here the bonding is disulfide bond. Okay, so this was simple. So when you got to know the bacteria cannot do one thing, so we make things easier for bacteria, and this is how we produce the humulin, which is the human insulin, by using the human gene only. This gene is a human gene only, and we express this in the bacteria. Later on, we joined it. Can we take insulin orally? Let me know. Can we take it orally? Yes. No, we cannot take insulin orally. The reason is it's a protein, and when you eat it, your pepsin is there, right? Your trypsin is there. It will destroy it. Aminopeptidase is there, so it will chop it into pieces, right? So that's why you never take insulin orally, you take an injection of it. Okay. All right. So this was about your genetically engineered insulin. Let's talk more about the other things. So this was how the Eli Lilly formed its first insulin, which is basically a therapeutic. Now, what are the other therapeutics that are there in the market apart from insulin? Or why are we making these first of all? So, what are the advantages of making these therapeutics like this? First is we can make these in a very large quantity. You can go for mass production. Second, they are safe and more effective because they are produced from what? They are produced from the human gene only. So they are more effective than the another, therapeutics formed from another animal. Another, since they are from our gene, they are formed from our gene only, so there will be no immune response. So this is how these our recombinant therapeutics are so much advantageous, right? So if I'm talking about recombinant therapeutics, what does it means? It means you are making a therapeutic with the help of a recombinant DNA. So this is how the therapeutic which is formed by these recombinant DNA technology, you call them as recombinant therapeutics. So in a worldwide, around 30 recombinant therapeutics have already been made like this way, and people out there are using. But in India, we are only marketing 12 out of these 30. Now, what are these 12? Let's see. You must have to memorize these. How many are marketed in India? How many globally already has formed? Okay. Now, let's see what are these other 12. Like human insulin, we have discussed streptokinase. Streptokinase is a clot buster. Erythropoietin, it helps in RBC synthesis. Hepatitis B vaccine that prevents hepatitis. Human growth hormone, human interleukin. This is like these are immunoproteins, interleukins. These are growth factors, granulocyte colony stimulating factor, granulocyte macrophage colony stimulating factors. These are certain growth factors that are used to, enhance the function or, in fact, the multiplication of these cells. Alpha interferon, again, this is the immunoprotein. Gamma interferon. Blood factor 8. Blood factor 8 is like a clotting factor and it is used in hemophilia. And follicle stimulating hormone FSH, and it is used in infertility. So this is how we have made these therapeutics, and there are a lot of other therapeutics that you can make, and you can use it against different diseases. So all these, when are we using all of these? Whenever there is a problem in a person. For example, human insulin, in diabetes. Streptokinase, if a person have clot and you want to burst it. What about erythropoietin? If a person is not having kidney, the kidney is producing erythropoietin. Okay, and erythropoietin is so much important for erythropoiesis. So at that time, if a person's kidneys are damaged, you cannot let RBCs to go down. Okay, so for that we need erythropoietin, so we can inject it artificially. Just like that, in all the immunological things, interferons, interleukin, they are used whenever you are giving immunotherapy, like in case of cancer. Okay, so this is how they are used against different diseases. So it's so good to go, right? Otherwise it would be a mess. People must be dying of all these diseases if these are not have been formed. Okay, all right, so say thanks to biotechnology. Next, moving further to the gene therapy, another application in the medicine part. So when I'm talking about gene therapy, what does it means? If I say, if a person is having a genetic disorder, that means a person's gene copy is damaged, or it has been mutated, or the wrong copy has came to that person, okay, during embryonic development. So what if I change that wrong copy with the correct one? The person's disease will go away. This is the only problem in genetics, that the person is having a wrong type of chromosome or a gene. What if we exchange it with the correct copy? The person will not face the problem. Okay, so this is what we do in a gene therapy. We replace the incorrect copy of gene with the correct one. Okay, so in 1990, which year? 1990. 1990, there was a girl child. She was suffering from SCID. What's a disease? SCID. Okay. The girl child was suffering from a disease known as SCID, severe combined immunodeficiency. So it's a kind of a disorder where a gene that is forming an enzyme, adenosine deaminase, is damaged. Okay, so what happened in this disease? The person's immunity gets deficient. The person have the low immunity. Why? Because the gene, which gene? ADA gene is damaged or not present, or wrong copy is there. Okay, ADA stands for adenosine deaminase. Now what happened is, guys, these adenosine deaminase gene, it's completely damaged. Okay, now if this enzyme is not formed, what will happen? The WBCs will not get mature. So this enzyme is very much important for maturation and multiplication of WBCs. Okay, if there is no ADA, there will be no mature form of WBC, and hence person will suffer from this SCID. So the immune system of that person is low, and the person can easily catch a lot of infections, and the person can eventually die as well. So we were thinking, if a gene copy is incorrect, let's perform gene therapy. But before gene therapy, what were the possible ways of treatment? So how can we treat the SCID? The treatment available was, first of all, you can inject ADA. ADA is that enzyme. But how long can you inject? Ultimately, the person is dependent upon it. Okay, the moment the person didn't get it, again the problem will appear. Second, there can be another thing, the bone marrow transplant. It seems simple that go for transplant, but it's difficult because they are not able to get this transplant tissue easily. Like bone marrow is not that easy. Why? If a person is donating the bone marrow, right, but it doesn't mean that it will match to the person who wants it. Okay, so there is a big challenge in this thing. And third thing is that we can go for gene therapy. We can go for gene therapy. Okay, so the therapy in which you are injecting the ADA, it is known as enzyme replacement therapy. Enzyme replacement therapy. Alright, guys. So these are three ways you can go for. So the best thing is gene therapy. So what we does in a gene therapy, see, if this is a bone of that person, in the bone there is a red bone marrow. Here is a red bone marrow, and in the red bone marrow these cells are getting formed and get matured. So what we do here is we extract the lymphocyte from here. Lymphocytes are WBCs only. It's a type of WBC. So what are these? These are lymphocytes. So we will extract the lymphocyte from a patient's bone marrow. From which bone marrow? Patient's bone marrow. Okay, so this is the bone of a patient. We extracted the lymphocyte. Now what we does is, with the help of a retrovirus, with the help of what? With the help of a retrovirus, because it's animal cell, we will give the correct copy of a gene. Now the correct copy of a gene will enter into these cell. Now we will allow these cells to divide. These cells will divide. After they divide, their number will increase, and now what we do is, these cells are having the gene. We will inject them back into the person's bone marrow. Okay, so we have taken the lymphocyte which were not having the correct copy. After we have given the correct copy, we will inject the lymphocyte back into the bone marrow. So how are we injecting it? We are injecting it with the help of vector, which is retrovirus. If you are giving the gene through retrovirus, and retroviruses have RNA, so here what type of a DNA is going? cDNA is going. What is a cDNA? Because retroviruses have RNA genome, so in our cell this RNA will converted into cDNA, and that is reverse transcriptase enzyme will help in this, and this process is reverse transcription. Okay, so because retroviruses are the vector that will throw the genome into it, but the genome is RNA, but you want the correct gene copy, and gene is a DNA. So what virus does is, a virus will convert the RNA into DNA, and this process is known as reverse transcription. and this occurs with the help of the enzyme which is known as reverse transcriptase. Okay, and here the DNA formed is known as a cDNA. C stands for complementary. Okay, so if someone asks you what type of DNA do you add, you will say cDNA. What is a cDNA? The DNA that is formed from RNA. Why we are putting the cDNA? Because our vector is retrovirus, and retroviruses cannot carry DNA, they can carry the RNA as a genome. Okay, so here reverse transcription is taking place, and the enzyme is reverse transcriptase. Okay, so this is how you can do the gene therapy. But again, since these cells, they are not immortal, they will die eventually someday. Okay, they will die someday. So what can be a permanent cure? If you do this gene therapy in the embryonic stage, for example, you know the baby is going to form, and in their history they have certain kind of genetic disorders. Okay, so if we undergo the amniocentesis, if the mother undergoes amniocentesis, and we get to know through that test in the early phases of pregnancy that the child may have this disease. So if we inject these cells into the embryo in the early stages, this can be a permanent cure. So gene therapy can be a permanent cure if done in embryonic stages. If done in embryonic stages. Okay, so this is how we have another set of treatments where you can correct the incorrect copy of a gene. So this was about the gene therapy. Let's move further and talk about the molecular diagnosis. This is another part of the application in medicine. So molecular diagnosis, what is a diagnosis? It is a set of technique wherein you will find out what is the cause of a disease. Okay, you are basically detecting the disease, you're detecting the cause of a disease. So diagnosis is a procedure, it's a procedure to detect the cause of disease. Okay, so for example, if a person is having symptoms, the person is having a lot of cough and breathing issues and all. Nowadays, what you call it? You call it, maybe a person catches the COVID virus, right? So in that particular situation, what you will ask a person to do? You cannot say you have a COVID straight away. Even the doctor cannot say. The doctor will ask that person to undergo RT-PCR. And RT-PCR is a type of a molecular diagnosis. It's a diagnostic test. So this molecular diagnosis is of two types. It is of two types. One is a conventional method. We also call them as traditional method. And another are the modern method. Okay. So the conventional method or the traditional methods are those which have been from the time like you were born, you were just listening to them. Have you ever heard of RT-PCR when you were so little? No, because these type of techniques are used nowadays much. In the later days, there were not such disease as much. Okay, RT-PCR has been, the labs has been increased while COVID. Otherwise, the labs were also so much less for this test because you don't need it much, right? So earlier test you must have heard like blood analysis, urine analysis, right? People give blood sample, check the diabetes, give the urine test or the stool test. So the traditional or the conventional method were basis on the blood or serum or urine or stool analysis. Okay, but these type of diagnostic tests they were like done when a person get the disease. Okay, if a person has diabetes, that will be reflected in the blood. That means you cannot find out in the early stages that a person may have diabetes in the future, right? You will only, or these tests will only let you know about a disease when a person has catch the disease. Okay, so this one does not tell about early diagnosis. All right, for example, if the child is not still born, it has not been born, it's in the mother's womb, and I want to see whether the child is maybe catch disease in some time or not, or later in the life or not, these tests will never let me know. Okay, but if I'm talking about modern techniques, they will let me know about the early diagnosis. whether that child can get the disease later on or not. Here basically we are talking about genetic analysis and all like that. Okay, so some of the techniques here are like PCR, ELISA, and autoradiography. So these are like high tech techniques. These are like high tech techniques which needs a lot of equipment, your labs, and some people who are experts in doing these. Though they are also experts who are performing these, but here these are more sophisticated than those. So now you must have heard about these in life, but these are a little bit different for you. Now, what are these techniques? Let's talk about in the detail. If I'm talking about the PCR, we have already done it in the previous lecture, guys. PCR is polymerase chain reaction. Polymerase chain reaction. So I will not be discussing it here because we have already done it in the previous lecture. But here I will discuss about the application part only. Where do you use PCR? If you want to detect a pathogen, if it is a pathogen of animal, pathogen of human, it's a pathogen of plant, you can detect it. For example, you have nice example, the RT-PCR. Why PCR is known as RT-PCR there? Because there the virus is a retrovirus. Fine, your COVID is a retrovirus and its genome is RNA. So we have to first convert it into the DNA, because in PCR you amplify the DNA, not the RNA, right? That's why it's the reverse transcriptase PCR. Okay, okay. Second thing, for paleontology, what is paleontology? The study of fossil. Study of fossils. Okay, so for in paleontology you can also use the PCR. Otherwise you can also use it for DNA fingerprinting to check the family disputes, whether this child is the parents or not, like father son relation, like that. Or during the crime scene you can check the samples of hair or blood, right, and then match with the suspect. So the which one matches, the person is a criminal, just like that. So there are certain other also applications of PCR. So next is ELISA. ELISA. ELISA is enzyme linked immunosorbent assay. Here assay is ASSAY. That means the detection. Okay. So what we does in ELISA, ELISA is based on antigen antibody interaction. It's all based on antigen antibody interaction. So it's the most common one used in the HIV infections, HIV AIDS. So when a person is having HIV AIDS, this is the test that is performed to check whether a person is having it or not. So what we do here is, we have a plate. We have a big plate of 96 wells. Wells are small, these holes, okay, like these. They have a space. So in these wells, we put the sample, and we put a lot of sample because we want to get, guarantee of the test. You just cannot do the test in one well and say, oh, eureka, the person is positive. No, not like that. You have to put sample in every well so that the probability becomes more, and there is a more guarantee that a person is positive or negative, for for getting it more ensure that yes, the test we have done is okay, it's not like false negative or something like that. So what we does in ELISA, basically we put samples here. Now what we do when we put samples, let's see. So in that well you have HIV antigen conjugated here. Okay, you already have this antigen here. We already have this antigen, which is HIV antigen. Now we will take blood sample of a person. We will take blood sample of a person. If a person is having HIV, the virus has entered, the antibodies will be formed. Agree? If a person is having, I am here assuming the person is positive. Okay, so if a person is having the you say the HIV in person's blood, the antibodies will be there and it will bind to the HIV antigen, because antibodies are specific to the antigen. Okay, now I know if a person is positive, the antibody will be of this kind, and now I have made an antibody against the antibody. I have made an antibody against the antibody, and I added an enzyme to it. So this is the one I have made artificially. This is the one which has came from person or a patient's blood, and this one was already present on my plate. My plate was, I purchased the plate by saying, give me the plate in which HIV antigen is there. Okay, so I added the person's blood, and a person's blood had this antibody. So it is patient's antibody. If a person or a patient is and HIV positive, it will show this antibody, and now my ready-made antibody will bind to it, and it has enzyme conjugated. Okay, because I want to see whether it is there or not. Otherwise, I will not be able to see with the naked eye that they have joined. I know a person is positive and this will definitely bind, but I cannot see it with the naked eye. So for that surety, I will add my ready-made antibody which have enzyme conjugated, and I will add a substrate. Because if a person is positive, this antibody will be there, this will bind, and substrate will be converted into product, and product will give a purple orange color. If a purple orange color is there, that means a person is HIV positive. Okay, if the person is negative, this will not bind, substrate will not convert into product, and there will not be any color change. All right, so this is how you can confirm whether a person is having HIV or not. So there are different types of ELISA. This is one type of ELISA. Okay, all right. Another types of ELISA will be doing in the human health and disease. Another thing is another type of diagnostic is autoradiography. So we have done PCR, we have done the ELISA, and now it is the autoradiography. What is autoradiography? So there is not a radio that will play a song, first of all. Whenever the word radio comes in the biology, basically it is talking about radioisotopes. Okay. And which one we are using here? Phosphorus. So you know there are certain radioisotopes of certain elements which can emit light. Okay. So what we do, what we are doing here, first of all, you need to understand that. Here we are detecting whether a person is having a mutation, have a have a incorrect copy of gene or not. For example, this is a normal gene of a person secreting an enzyme. A person is living happily. But if any mutation occurs in a gene, the person will be diseased. So in a diseased person, the same gene when gets mutation, for example, the mutation is like a delete, is a deletion. Okay, and here a G, it subtracts. So the person's gene will be like this. Okay, just like this. All right. So here we are showing that a person's G nucleotide has been, guanine has been removed by the deletion by mutation. So this is a normal gene and this is a diseased gene. This is a diseased gene and one nucleotide has been deleted during mutation. Okay, so I have to check whether a person is having the genetic mutation or not. A person came to a doctor and told about the symptoms the person is facing. Now the doctor will see by these all symptoms, maybe the person is having that genetic disorder. The person will ask a sample of cells of a person, that patient, and those cells will then undergo the autoradiography. So there you have extracted the gene, because the person was on a suspect of a particular genetic disease. So we will extract that particular gene only. Now what we did, we made a probe. We made a probe. I know if a person is having a normal gene of this particular disease, if it is not a diseased person, the probe will bind to it. We have taken a radioactive probe. We have taken this segment of RNA, of DNA, and that's radioactive. So this is radioactive DNA. So any radioactive DNA or RNA, it can be RNA, is known as a probe. It is known as a probe. Okay. So what we did, we asked this probe to bind to both these genes. Okay. So for example, first of all, there are not both the genes. We are taking the person is normal. There are two different cases. Okay. If I'm thinking that a person is normal, is not having any disease, this probe will definitely bind to it. See, this is bind to it. and when you send some light to it, and you will see a particular type of light like green. Okay, and this will tell you that a person is not diseased, it's normal. If a person is diseased, probe will not be able to bind. Why? Because probe is like this, it will not be able to properly bind. For binding, we need that a proper sequence should be there. A proper sequence should be there. Or to make it more easy, let's see the deletion has or another mutation has taken place, like GGACCC. That's the normal gene. The A is there. Now the probe will not be able to bind, right? Because the probe is CCCGGG. Can it bind easily? No. At the place of C, there should be T. So there will be no binding. If there is no binding, there will be no emission of light. And if there is no light, that means the gene is mutated. So this is how you can find out a gene is mutated or not. If a person is normal, there will be emission of light because these are radioactive phosphorus. Okay, so when you give any type of a high energy light to it, it will emit the radiation. Next, if a person is diseased, if a person is having any disease, there is not a set sequence that can bind to this probe. Hence, there is no binding, no emission of light, and a person is diseased. If a person does not show a light there, that means the gene is mutated. If it shows a light, that means the person is normal. So this is one of the another high-tech technique that you can use to check whether a person is diseased or not. And having a which disease? Genetic disease. All right. Moving further to the next, what is that? Transgenic animals. Now, what are transgenic animals? Yes, there is a very cute animal that you can see. And yes, I have done a lot of experiments on them. So I have a lot of, love and respect towards these mice. Okay, when I was doing research, I did on the albino mice, Babsy mice. They are so cute, I tell you, they are literally so cute. So, what first of all are transgenic animals? Animals in which the gene is manipulated. That means you have added a gene or you have deleted a gene. Okay, so first of all, what are transgenic animals? Animals which have had their genes manipulated. Their genes manipulated. Either you have deleted a gene or you have added a gene. Out of all the animals, 95% transgenic are mice. That's why I have put very nice images of them. So 95% are mice. Now you must be thinking, why are we making so much of transgenic mice? Or ma'am, why do we why were you doing those experiments on the mice? I was trying to do a research on a medicine for leishmaniasis. Okay, so why are we making these mice? Because they are, first of all, mammals. They are near to us, right? Second thing, their physiology is little bit similar to us. And why are we manipulating their genes? Depending upon the use you want. For example, if I want to study cancer, I will induce cancer in that mice. I'm manipulating a gene. That's how this becomes transgenic. Or if a person is having a genetic mutation or a genetic deletion, and this is how a genetic disorder occurs, I will also do that deliberately in a mice and make a transgenic mice with that particular disease. Okay, so that I can test my medicine on it. I just cannot research and make a medicine and tell a human to eat it. Fine, so you need to do it on a certain animals later on, so that it can become possible for humans to consume. Okay, all right. So otherwise, out of all the animals, 95% are transgenic mice. But what are the others we have made? We have made transgenic sheep, rat, rabbit, monkey, goat, cow, etc. So we have also done it in other animals as well, but the maximum are mice. Again, why? Due to research. Okay. So if we are talking about the transgenic animals. Let's first talk about, all right, this cow. Who is this cow? This is Rosie. So let me introduce boys and girls to Rosie. Rosie in the house. So Rosie is a transgenic cow. Okay. So, how she was so much helpful? We added a gene, and this gene was producing a protein alpha lactalbumin. Alpha lactalbumin, 2.4 gram per liter of milk contains alpha lactalbumin of a Rosie cow's milk. Okay, the Rosie's cow milk contains around 2.4 gram per liter of the milk, this protein, which is alpha lactalbumin. Okay, so we added a gene, this was producing this protein. And why this protein was so much important? If you give a normal cow milk to a newborn and you give a Rosie cow's milk to a newborn, the newborn will be having a, you know, beneficiary, will show the beneficiary result of a Rosie's cow. Why? Because it is rich in protein. Okay, so this is how they made it a transgenic cow. That's good. So if you want you know, healthy diet, you can just add a gene and the animal will produce it. Fine. Okay, but some things are unethical, you cannot do that. Otherwise otherwise, what are the various uh, importance of transgenic animals? What is the various importance of transgenic animals? Let's see. If I'm talking about transgenic animals, where they show us the benefits, one field is the medicine, another is the agriculture, and then we have industrial application of transgenic animals. Okay, how? If I'm talking about medicine, how transgenic animals are so much important? First thing first, these animals help us to know about the normal human physiology. If I say most of our organs are similar to the mammals, like mice. So if you study the mice normal physiology, you can also get to know about our physiology. Yes or no? So the first application in the medicine is to study normal human physiology, that how our body functions, they work and all. Second thing, to study the diseases, study of diseases. Okay, that how did we get cancer, how do we get this genetic disorder? So by inducing those animals to get disease, we can study the diseases. Also, we can test the drug and vaccine testing, we can also do this. Otherwise, also for biological products, for example, we have made Rosie. And Rosie was producing a lot of this what do you say, the milk. Okay, apart from this, we have also made a transgenic sheep. And its name is Tracy. Okay, how cute name. The cow was Rosie and the sheep is Tracy. Okay, so this Tracy was producing alpha antitrypsin. What is antitrypsin? So it is used in emphysema. You must have heard of this disease, emphysema. I have taught you in the breathing and exchange of gases. It's a disease where if a person smokes a lot, the lower respiratory tract gets damaged, reducing the surface area, hence no exchange of gases and a person can die. Okay, so what happened in this disease? The wall of a respiratory tract, it started secreting a lot of trypsin. And this trypsin enzyme, it digests the fibers. When it will digest the fibers in the wall of respiratory tract, there is loss of elasticity, and hence there will be collapse of the tract. And when it's a collapse, you cannot breathe. Fine. So we found out that this trypsin is the one which is creating the mess here. Okay, so we formed something which will block the trypsin, and that is antitrypsin. So now the question was, who will produce antitrypsin? So we added a gene of antitrypsin in the sheep Tracy. So Tracy is producing alpha antitrypsin with the milk, and this is used in emphysema. Apart from emphysema, what other diseases we have? We are working towards that should get the person should get benefit in. So that is phenylketonuria and cystic fibrosis. So we have also made the transgenic animals for these diseases as well, apart from the emphysema. Okay, all right. So now you got to know about the biological products. One, we have made the milk rich in protein. Another, we have made the alpha antitrypsin. What about the agriculture? In agriculture, for breeding, the breeding is a very, difficult task in the agriculture. Conventional breeding is so tough. You want one animal, then the another, and then let them mate, and you don't know whether you will get the desirable product or not. So for that, what if we switch it with the MOET? MOET is a, thing of biotechnology only. Yeah, during MOET, you are transferring the embryo. Okay. So what if you just change MOET a little bit? You let the eggs and sperm meet in an artificial condition. There you also add a particular gene, and that animals get a good gene, and later on the embryo is formed, and then you will put that embryos in the surrogate mother. It is quite simple. Yes. So to avoid this normal breeding, we have the applications here in the agriculture, because breeding conventional is tough. So there we need the biotechnology. Also to have better variety of plants. Okay. Also to have better variety of plants. And definitely for high yield. So for all these benefit, what we need? The biotechnology in the agriculture. Next, we have industrial application. So in industrial application, there was a company known as Nexia Biotechnology, which is in Canada. They added spider web genes. They added spider web genes in a goat. Yes. Now, goat will secrete the web proteins in milk. These web proteins, they are like a silk protein. These are like silk protein, and by extracting this silk protein from the milk of a goat, they produce tennis rackets, army uniforms, and even the sutures. Sutures, you understand? When you get on the skin, when you get a deep cuts, the doctor give the sutures, right? So this is how they have, they brought the biotechnology to at the industrial scale. See how good, how nice, right? So these are various applications of your, different types of biotechnologies application, and specifically the transgenic animals application in different field. So what can be the benefit of agriculture and transgenic animals? So here I am saying there's a better variety of plant. Also there can be a better variety of animal. That means a animal can be high yielding, disease resistant. All right. Okay, so this was about the application of transgenic animals in the different fields of life. Let's talk about the next issue, that's ethical issues. Again, ethical issues. Let's talk about them. So now there are three terms in the field of biology. In fact, you will get to know. These are bioethics, biopatent, and biopiracy. These are very much important when you are doing certain research. Okay, because while you are doing a research, maybe you will get across a discovery or invention, and if you are the first one to do it, you will get its rights, and that rights are known as biopatent. Okay. Okay, let's just talk about one by one. First of all, we'll talk about the bioethics. Ethics means the rights. Okay, the set of standards, basically, not just the right set of standards. So there must be a control on what research is going on, otherwise we will make something havoc that will, can create a lot of mess in the society. Okay, for example, somehow if there is no set of standards that you can do this research and you cannot do this research, or if there is no control over the people on the research, maybe people will start doing different stuff and harming others. So there are certain bioethics that you can go you cannot go beyond this line. You have to stay up to this. You have to do research only whatever is covered in this particular segment. You cannot go beyond it, because then it will be unethical. So there are certain societies that give the set of standards to control the research in the field of biology. In the field of biology. So, since I was talking about there are certain organizations in every government. So, in India, we have a government organization which is GEAC, Genetic Engineering Approval Committee. Do remember its full form, it's important. So, this committee will see whether this research is valid, you can do it or not. For example, if I want to research on something, I want to start my PhD. So, I will write everything on some papers that this is what I'm going to do in my entire PhD. This is my aim. This is what I'm trying to find. These animals I will be using. So if they find it ethical, they find it, yes, you can use these animals, yes, you can do this type of research, they will give me a big okay. If there is something unethical, for example, I wrote I will do experiment on this animal, but that animal is endangered, then I will not get the big go. Okay, so these are the rights which is taken care of by this committee in India. Okay, why there is a need of all this? Because if we started doing experiments on human, or we make some organism which is half human like and half dog like, so will there be any humanness thing left in this world? No, you will come out with another species which may harm the human species, and later on maybe we will get vanished one day. So that's why it's always important to get a control on what we are doing. Okay, next we have biopatent. So for example, as I've told you, I was doing a research, and somehow if I get to know, yes, this is a drug and this is so much effective on this particular disease, I will get a patent of it. Why? Because before me no one has does this. Okay, this is known as novelty. It's completely new, no one knew about it. Non-obviousness, that means no one knew about it. Utility, it's important, that means it's important because people who are suffering from that disease will be cured. So biopatent, these are the rights given by the government only to the person who has done the invention or discovery. Okay, it can be for a lifetime, it can be for a shorter period of time, but these people will only get the patent of that particular research or invention if that invention is new, that means it has novelty. Second, it's non-obviousness. It doesn't mean, for example, if out there in the western world someone will say use turmeric, it will help in wound healing, but in India we already know about it, right? So that's absolutely it's absolutely obvious, so you will not give patent of it. And third one is utility. It should be of a use. I invented something and it is of no use, I will not get a patent. Why? Because it's not good for human welfare or any other animal or even the environment. Okay, so patent are the set of rights given by government. It can be state government or central or national government to inventor or a person who does or who did the research or who did the discovery for public disclosure. For public disclosure. So what does this biopatent should have? It should have three properties. One, novelty. It should be new. Non-obviousness. And the last one, utility. Okay. Now come the biopiracy. Biopiracy is like some people are thieves, and they are without knowing or without consulting you, you're taking something out of you. Okay, so biopiracy, this is a misuse of bioresources of a particular country who's rich in resources by multinational companies from a country which is rich in resources without telling them, without proper authorization. Okay, and not giving them any right or any compensation out of it. For example, if someone from the another country come to India, take our plant and start claiming that I have done this research, fine. Is it ethical? No, this is not ethical. You first of all, you have to tell the country you are taking a plant, though they will not allow that person to get it from the immigration. But you have to tell the company you have proper, every paperwork done. Only then you can do it. Fine. So biopiracy is the misuse of bioresources by multinational companies, by multinational companies without proper authorization of the people concerned. So we have faced these biopiracy issues earlier. Let's discuss what were these. First was in September 1997. Texas in America, there is a company known as Rice Tech. What they did, they took our basmati rice variety, basmati rice. They crossed it with their variety, their rice variety. Whatever is formed by this, for example, X rice variety is formed, they took a patent of it. They took a patent of it. Okay, which is not ethical. Why? They didn't ask about our basmati rice. In India, there are 27 documented rice basmati varieties, right? And there are around 2 lakh varieties of rice that are grown in India. And it's also mentioned in our old books and everything like that. Then how can any other country take our gem out of our country? So it's not ethical. So we took this issue to the World Trade Organization, and then some of the rights are taken back. Again in May 1995, University of Mississippi. University of Mississippi. This university published a paper about wound healing properties of turmeric. Wound healing properties of turmeric. And you know what is turmeric in Hindi? It is known as haldi. And we already know, everyone drinks the haldi ka doodh. Haldi's milk, turmeric's milk. During whenever we get some injury, or your mothers used to give, and it's so good to our health. It has anti-inflammatory properties. So how can they publish the paper? The thing which we know from the ages, right? Again, neem have properties which is insecticidal properties, right? So this this neem's patent was taken in 1996 by Germany's, right? There was a company known as W.R. Grace and Company in Germany. They took the patent of neem. And what was the patent? The patent was on that this neem is having the antimicrobial property. Okay, the fungicidal use of the neem. But we know from ages we are using face wash of neem. What are the people saying? Right? So we already know that it has antimicrobial properties. So how can they take the patent? So by this we revolted and we took their rights back, and this is how we protected our country's resources. All right. So this was all about this chapter. Let's solve some of the questions, guys. First, in RNAi, the genes are silenced using double-stranded DNA, RNA, single-stranded DNA, single-stranded RNA. You know how was RNAi initiated? When there was formation of double-stranded RNA. So what will be the answer? Second. Okay, next. Use of bioresources by multinational companies and their organizations without proper authorization from the countries and people concerned, without compensatory payment, is your biopiracy. What is a biowar and what is a bioweapon? In biowar, the bioweapons are used. What are bioweapons? These are certain microbes or disease causing organisms. One country will send the disease causing organism, in the world war, in the earlier time the smallpox virus was used once. to kill the people of the another country. So that's a bio war in which your biological weapons are used. Bioethics, these are set of the standards that you keep in mind while doing the research. So answer will be 3. Next, which of the following is commonly used as a vector for introducing a DNA fragment in human lymphocyte? So, which vector did we use during gene therapy in a 4-year-old girl child which was suffering from SCID? Definitely retrovirus. Human lymphocyte, first of all, we are animals. So it's quite simple. In animals, what we use? We use the retrovirus. TI plasmid is used in plants by agrobacterium. PBR322 in the bacteria. Phage also in bacteria. So you just have to see what is the host. If you know it, you can get a correct answer. So answer is 1. Next. Which of the following is commonly used as vector? Okay, that's repeated, I guess. Okay. Which of the following is true for golden rice? It is vitamin A enriched with gene from daffodil. It is pest resistant with a gene from Bacillus thuringiensis. It is drought tolerant, developed using agrobacterium vector. It has yellow grains because of a gene introduced from a primitive rice variety. Okay. So most of you will get confused in option 4 and 1. Okay. So the students were quite confident. They will directly read the question, and in their mind, by reading first statement, they have decided the answer is 1. It's absolutely correct. Okay. The second one is saying it's pest resistant. No, it is about the BT cotton, what they have written. Second, it is not a drought tolerant. It was a nutritional, high nutritional rice with vitamin A, which you have taken from daffodil. This is incorrect because they are telling that the gene of beta carotene is taken from another rice variety. No, no, no. You have taken the gene from the daffodil, you have added it in the rice variety. So the answer is 1. Okay. All right. Again, I guess. Okay. BT toxin kills insect by inhibiting protein synthesis, generating excessive heat, creating pores in the midgut of epithelium, and option 4 is obstructing a biosynthetic pathway. You know how that toxin was killing the insect? It was entering into the midgut. In the cells, it was creating pores. It was creating pores. As a result, the water enters into the cell and cell burst. So, what will be the answer? By creating pores in the midgut. So, answer is, so they were asking about the BT cotton. Okay. All right, so this was all about biotechnology and its application. Other chapters I'll be covering, and I'll be giving you most other, like, I think, human health and disease next chapter. So till then, you have to prepare the biotechnology very well, guys. Yes, keep on working really, really, really hard, and I'm there with you always. Just keep on working hard and let me know that you have finished it or not. All right, bye bye. Take care. Lots of love.